So what did we learn in till our previous class, ma? We have dealt with monohybrid, dihybrid, exceptions of monohybrid cross. What are the exceptions of monohybrid, ma? Exceptions of monohybrid cross means what are the exceptions for law of dominance? Tell me.
Independent, incomplete dominance.
Very good.
Co-dominance.
Superb.
Multiple.
Great. Okay. Can you tell me the examples of incomplete dominance?
Snapdragon.
Snapdragon, flower color, antirrhinum. Co-dominance? Blood grouping, human blood grouping, ABO blood grouping. Multiple allism. Think. Starch synthesis. ABO blood grouping also. Pleiotropy. Pleiotropy examples. Starch synthesis. Starch synthesis is not for multiple allism, it is for pleiotropy. Multiple allism, it is only ABO blood grouping. Okay. So you have starch synthesis and phenylketonuria disease, skin depigmentation. These and all are the examples of pleiotropy. Fine, right, beta? And dihybrid cross, it's numericals and learning phenotypic ratio, genotypic ratio for calculations. Okay. Exceptions of this, what we have learnt, exception of law of independent assortment. Exception of law of independent assortment. Polygenic inheritance, right? And then we have looked into formulae, applications. Okay. Fine, right? Chromosomal theory of inheritance. Okay. And gene distance, chromosomal mapping by Alfred Stutward, experimental verification for chromosomal theory of inheritance. So first half of the cinema is done. Now second half. In this second half, what we are going to learn is sex determination. Okay, 3 modes of sex determination, followed by disorders. Okay, genetic disorders, we have 2 types of genetic disorders. Those disorders which follow Mendelian pattern of inheritance comes under Mendelian disorders and chromosomal disorders, followed by pedigree analysis. This finishes the next half. So here if you see beta, first is sex determination. What is sex determination? Determining the gender or sex of an individual in the early stages of the life. Okay, for any organism it might be. What is sex determination? Yeah, determination of sex of an individual at early stages of life. Fine, right? So it is different for different organisms beta. We have environmental factors which affect the sex determination, that will decide what will be the gender in the early stages. Mostly we will know the genetic mechanism and haplodiploid mode. We will discuss examples one by one. So environmental factors, best example I can take is turtles. Fine, right? Turtles at the time of their egg hatchling, if they are hatching at low temperatures, that turns out to be a male. Okay, if egg is hatchling at high temperatures, that turns out to be a female. So here environment is deciding the gender or sex of the individual. Fine, right? And genetic mechanism. We know females will have certain set of allosomes, males will have certain set of allosomes. So based on the number of allosomes or type of allosome, whether it is in male or female, based on that differentiation we have genetic mechanism of sex determination. As such we have 2 types beta. First is male heterogamety, and second will be female heterogamety. Fine, right? Male heterogamety and female heterogamety. Male heterogamety is again of two types. Fine, right? We have XO, only one X chromosome, okay, not two sets will be there, and one is different types of allosomes in the male like XY, either having one set or two different sets. Okay. Next is female heterogamety beta. Female heterogamety will be ZZ, ZO, either one set or two sets, ZZ, ZW. Fine, right? Next is what? Haplodiploid mode of sex determination. This you will see it in honeybees. Honeybees, where complete set of chromosomes, that is diploid set, is observed in females, whereas half the set of chromosomes or haploid set is observed in males. Okay? This is haplodiploid mode of sex determination. Diploid condition or complete set of chromosomes are present in females, or haploid set of chromosomes or half the set of chromosome is present in males. Clear till here? Any doubt anywhere? No, right? Just take down briefly with the examples. We will move on to genetic mechanism of sex determination. Important, Abdullah. For this also examples are important. We will get match the following type of questions. Once it is done, let me know. Now, here we will go with chromosomal basis of sex determination or genetic mechanism. First, what is there, ma? Male heterogamety. Here, if you say, as I discussed, you have either half set or complete set, but two different. Same the case with female heterogamety in the allosomes. Either you will have half the set or complete set in the females. Here it is in males. As such, you have male heterogamety and female heterogamety. Now, we will see sex determination, that is male heterogamety. As I told you, first examples are important. Beta. So, half the set of chromosomes, that is XO, male type of sex chromosomes, these allosomes, is observed in bugs, cockroach, and grasshopper. Fine, right? Bugs, cockroach and grasshopper. Here we go. Can you see here? Total how many chromosomes? 21 chromosomes are there. Both the sides. See, male and female. 21 chromosomes. Here 22. Here only 21, absence of one chromosome. See, 10 and 11. So, gametes, if you see, 10, 11, total 21 chromosomes are seen in male, 11, total 22 you can see in the female set. Simple as it is. Don't get confused between autosomes and allosomes. Okay? Here absence of one allosome is deciding the gender. That absence of one allosome is there in males. That is why you address it as what? Male heterogamety. Did you take the examples? Identification and examples, enough better. Fine. Next you see sex determination in Drosophila. Same the case you observe here. Drosophila melanogaster, that is fruit fly, it has 4 pairs of chromosomes. Total it has 8 chromosomes in diploid set. You see male and female, 4 pairs, okay. But here in the allosomes, one pair you have XY, here you have 2X chromosome. Based on the combinations, XY will be males, XX will be females, as simple as it is. So, this is XXXY type of sex determination observed in humans and Drosophila. Humans and Drosophila. Clear? Female heterogamety, that is ZW, ZZ. Fine, right beta? So, here ZW heterogamety is observed in females. Here you can see ZZ and ZW. Based on the type, ZW are turning as females, both homozygous condition is giving male offsprings. Fine, right? So, this type of sex determination is observed in birds, fishes and reptiles. Birds, fishes and reptiles. Next, ZO, ZZ type of sex determination. ZO, ZZ type of sex determination. Here you see ZO type, only one set of chromosome. Okay. So, Z along with autosome, ZZ will give rise to male, ZO will give rise to female, half the set of chromosomes. This you will observe in butterflies and moths. Have you taken the examples of everything? Cool. Done, right? Now, haplodiploid mode of sex determination. I told you females will have complete set of chromosomes, like females diploid set of chromosomes is 32. Fine. Males will have, as I told you, half the set of chromosomes, that is 16. Example, drone you take. Okay. This undergoing mitosis only, they will produce haploid set of chromosomes again. Whereas, this as it has complete diploid set, this will undergo what beta? Meiosis or reductional division, and half the set of chromosomes will be produced. Okay. Whereas, one set will turn to drones or males, half the set of chromosomes. The other half set will undergo gametic fusion, forming out 32 chromosomes that turns to females. So, here if you see in this generation you can see females, and in this generation. Okay. This again undergoes meiosis, forming 16, again here male chromosomes, gametic fusion will result in females. So, males are produced without fertilization, yes or no? Without fertilization. Females are with gametic fusion or fertilization. This is saying like males are having grandfathers and grandsons, but no fathers and sons. Here in haplodiploid mode of sex determination, males will have grandfathers and grandsons, but no sons and fathers. Okay, this is haplodiploid mode. Complete set is for female queen and half the set for males. Clear, right? Sex determination concept is clear, Noma? Now we will move on to disorders. Now, genetic disorders we have two types, Mendelian and chromosomal. First you have to know what are Mendelian disorders. The disorders which occur because of gene mutations. Gene mutations. So you know the condensed form of a chromosome, the condensed form of a chromatin is a chromosome. Yes or no? Chromosome is the condensed form of chromatin. Here you have the DNA, right? Over which genes are present. Fine, right? So mutation in any one of the gene will cause what? Gene mutations. These gene mutations will occur through either transition or transversion. Either deletion, insertion of any one of the gene will result in what? Gene mutations. Occurring gene mutations, these are inherited to the next generations following Mendelian pattern of inheritance. Mendelian pattern of inheritance. Clear, ma? Fine, right? Gene mutations, they will occur and they are inherited to the next generations following Mendelian pattern of inheritance. Next is chromosomal disorders. Okay, chromosomal disorders occur through chromosomal aberrations, either change in chromosomal number or structure. Either change in chromosome number or chromosome structure. If you got a brief idea on this, then we will move on to each concept in detail. Okay. Now, look at here Mendelian disorders. Fine, right? So, before entering into the Mendelian disorders, I have to make a clear note on, just a second, yeah, Mendelian disorders. You take down this flow chart beta. This can be autosomal or allosomal. Autosomal again can be dominant or recessive. Fine, right? Allosomal can be X-linked or Y-linked. This is foundation for your pedigree analysis also, listen carefully. So, here you see dominant mutation means dominant form of inheritance. It can be expressed, dominant autosomal means it can be expressed both in homozygous condition and in heterozygous condition. Best example will be myotonic dystrophy, muscle disorder. Recessive diseases expressed only in homozygous recessive condition, yes or no? Homozygous recessive condition. Many a times you got questions in your NEET exam on autosomal recessive disorders. If you see, we have phenylketonuria, sickle cell anemia, thalassemia, cystic fibrosis. X-linked UC. X-linked again we have dominant or recessive traits. X-linked dominant, X-linked recessive. X-linked dominant is expressed both in the homozygous and in the heterozygous condition, whereas X-linked recessive is expressed completely in homozygous recessive condition. Major important examples, many at times asked in NEET PYQs on recessive traits, recessive X-linked recessive disorders like color blindness, and hemophilia. Okay. Y-linked disorders, these are directly transmitted from father to son, like bald-headedness, polydactyly, these and all will be Y-linked inheritance. Okay. Make a clear note on this. If you are having any doubt, just ask me. Take time, but note everything, ma. Good. Now we will look into each pattern of disease in detail. First, color blindness, X-linked recessive disorders. Color blindness, of course, as the name itself is saying, X-linked recessive, it is expressed only in the homozygous condition. If it is expressing in the females, it should be in homozygous recessive condition. If it is heterozygous condition, it is addressed as carrier female. Fine. Next, in males, if you see beta, if disease is on X chromosome, then definitely it is inherited. Fine. This will be the pattern of inheritance in both females and males, pattern of disease. Now you see, what is color blind disease actually? This is X-linked recessive, as I told you. There is mutation in one gene on the X chromosome which will show the effect on the cones of the eye, where the patient or the affected person will not be able to distinguish between red and green colors. For example, this is the normal vision, okay, normal vision who does not have color blindness. But when red and green colors are shown, this will be the vision of a person who is having color blind, and the effect will not be there in the heterozygous condition. They will have normal vision only in the homozygous females, and X chromosome affected males will have this color blindness. Clear, right? The disease is unable to distinguish between red and green colors. Cones will be affected. Fine, right? So here you see, color blindness is X-linked recessive trait caused by deficiency in either red or green cones in the eye. This causes inability to distinguish between red and green colors. This deficiency is caused by mutation in one of the X chromosomes gene. Fine, right? But they will follow crisscross pattern of inheritance. This affects 8% of males and just 4% of females. Only 4% of females, because if it is in heterozygous condition, it is not showing any effect. Just 4% of females. This is important. Note down now. This you can analyze easily, X-linked disorders and what is the approx stats of effect of this disease on males and females, just note down. then we will see all the possible crosses, and we will know the probability of the offsprings with the disease, without disease, and carriers. Done. Now you see, we will see 4 types of crosses. First, where affected female is crossed with unaffected male. Second type of cross, affected female with affected male. Okay. Third, you see beta, carrier female with unaffected male, carrier female with affected male. Now look at the probability of the offsprings. How many are affected? In this cross, how many will be affected? Will you tell me? What are the possible outcomes? So, how many are affected? If at all in your question they are asking you, mentioning the progeny, what is the probability of affected progeny? Then your answer should be 3:1. Okay. Or else 75% of progeny are affected and 25% remain as carriers. Okay. 75% affected, 25% carriers. This should be the probability. If they are specifically asking you in between males and females or daughters and sons, what is the probability of affected sons? 100% of sons are affected in this pattern of cross. How much percent of daughters are affected? 25%. Clear? If daughters and sons are asked, this should be the thing. If progeny is asked, this should be the ratio. Clear? Understood, ma? Next you see, this one you only have to tell me. All the offsprings are affected. %100% of the progeny is affected. This one you see, tell me. Fine. 25% of progeny affected, 75% unaffected. How many carriers? For unaffected you have to include carrier also. Now this one you see, what are the possible outcomes? How many are unaffected? Half, 50%. Fine. How many carriers? 25%. How many affected? take down. Just take it as homework, keep 4 headings, identify how many carriers, unaffected, and in between males and females also. Any doubts though, you can watch the recorded later. Now, hemophilia. First let me brief you out about this disease. Hemophilia is because of absence of one blood clotting factor. Okay, we have 2 types of hemophilia, hemophilia A and hemophilia B. Fine, right? Hemophilia A or B, it is because of absence of one blood clotting factor which will not allow the blood to clot in any wounded regions or as such. Blood will not be clotted. Okay, as such it will it is also addressed as bleeding disease or bleeder's disease. Fine. Hemophilia A and hemophilia B, these are the 2 diseases. And how is this disease inherited? X-linked recessive disease only. Okay. First it is identified in the pedigree chart of Queen Victoria family, okay, wherein all the descendants of that family are affected with this. Fine, right. So, this shows transmission from usually carrier female to the male progenies, because X chromosome is inherited. Affected X chromosome is transmitting to the male progeny, so it is affected. And here this is due to the presence of defective form of blood clotting factor which will not allow the blood to coagulate properly. Here you can see this is the normal blood vessel. If bleeding starts, hemophilia, this clot will be incomplete fibrin clot, and completed fibrin clot will be this. As the incomplete fibrin clot, blood will flow out or ooze out completely. Fine. So, here if you see, we have 2 types. One is due to the low amount of clotting factor, that is anti-hemoglobulin protein. Anti-hemoglobulin protein. Next is hemophilia B. This is also called as Christmas disease. This is due to the absence of clotting factor 9. Okay, clotting factor, that is plasma thromboplastin. Fine. So, this is the chart, if you can see, from carrier female, how long the disease is transmitted. Here you can see one male got affected in this generation. Next, here in 3rd generation you can see again males who are affected. In next generation also you can see affected males from carrier females in every generation. Fine, right? Sickle cell anemia. Now we are starting with autosomal recessive disorders. Sickle cell anemia is because of the absence of one gene, that is hemoglobin beta gene on the chromosome 11. Okay. Because of the absence of this one gene, usually the red blood cell shape will be round and biconcave. Okay. Because of the absence of just this one gene, that is hemoglobin beta, okay, this gene, it is making the shape of hemoglobin turn from round biconcave to sickle shaped red blood cell. Round biconcave to sickle shaped RBC it is making into. Fine, right? Sickle shaped RBC. Because of this, what is the usual function of RBC? To transport oxygen, right? Okay. Because of the change in shape, it cannot associate with oxygen properly, thereby oxygen transport is affected. Okay. As we have discussed, the effect is on hemoglobin beta gene. In hemoglobin beta gene, what happens is, just because of change in one nitrogen base, that is GAG on the mRNA, GAG is turned to GUG, okay, RNA, where A is turned to U. GAG will be coding for what amino acid? Glutamine. Here GUG is coding for what amino acid? Valine. This itself is bringing out the disease. Okay. Where is this changed? In the mRNA. That is, normal hemoglobin DNA is having CTC. It is coded to mRNA as GAG. Whereas mutant hemoglobin DNA is having CAC, it is coded as GUG. That is how the effect is seen. So, here if you see, the deficiency of RBCs is due to replacement of mutation from glutamic acid to valine at which position? 6th position. That is why it is addressed as hemoglobin beta gene on 6th position. Globulin change is at which? 6th position. When amino acid in the globulin protein is substituted, this is a point mutation, also addressed as qualitative mutation. This is because of transversion, okay, where the purine adenine is exchanged by what? Pyrimidine. Likewise. So, here pyrimidine, thymine is a pyrimidine, right? It is exchanged by purine, that is adenine. It is transversion. So, it is what beta? It is a point mutation, only single gene is mutated. Okay, it is the result of transversion, where T is, that is pyrimidine, is transversed to purine, that is adenine. Fine, right? Point mutation, transversion, and as mutation in a single base is resulting this effect. This is what? Qualitative. Qualitative. Fine. Now, here you see, when an amino acid of the globin protein is substituted, the consequence is single base substitution at 6th position of beta globulin gene, that is GAG to GUG. The mutant hemoglobin molecule undergoes polymerization under oxygen tension. Oxygen tension will be created because it is not transported properly because of change in shape of sickle, change in shape of RBC from round biconcave to sickle shape. Fine. Take down the points quick. Then we will move on to thalassemia and phenylketonuria. Phenylketonuria, here if you see, this is inborn error of metabolism beta. Okay. This results due to decreased this results in decreased metabolism of amino acid phenylalanine. I will tell you, see, first phenylalanine, to show its functional activity, it should be converted to tyrosine. Okay. This is done by the enzyme phenylalanine hydroxylase. Absence of this one enzyme, if this enzyme is absent, phenylalanine is stored in the form of phenylpyruvic acid, and this gives multiple effects, okay, multiple effects like mental retardation, poor absorption in kidneys, frequent micturition, skin rashes and hyperactivity. All these effects are shown because of the absence of one gene, that is phenylalanine hydroxylase. Fine, right? This is how the effect is observed. This is because accumulation of this in brain will result what? Mental retardation as well. So, as one enzyme affecting multiple functions, we will learn this as an example of what? Pleiotropy. Clear? Autosomal recessive disorder, and it is also an example of pleiotropy. Next, thalassemia. Sickle cell anemia is a quantitative disorder, yes or no? Sorry, qualitative disorder, yes or no? But thalassemia is a quantitative disorder. Okay, thalassemia is a quantitative disorder. This is also autosomal recessive. You know, hemoglobin in its quaternary structure, it has 2 alpha globulins and 2 beta globulins. 2 alpha and 2 beta will be there. 2 alpha subunits and 2 beta subunits. So, here alpha thalassemia and beta thalassemia. If alpha globulins are reduced in their synthesis, if you can see reduction in the synthesis of alpha globulins, that results in what? Alpha thalassemia. Beta thalassemia results, this is because reduction in the synthesis of beta globulins, right. So, here you see thalassemia, this is production of alpha globulin is affected over here. This is because of the 2 closely linked genes, this HBA1 and HBA2 are affected on the chromosome 16. Mutation or deletion on any one of the chromosome will result in the reduction in the synthesis of alpha globulin. What you have to remember here, which chromosome, what are the genes on that chromosome are affected. Alpha thalassemia, what are the genes ma? HBA1, HBA2 on chromosome 16. Beta thalassemia, if you see, production of beta globin chain is affected. This beta globin production is controlled by single gene, that is HBB, this is single gene on chromosome 11, this is on the chromosome 11 of each parent. Mutation of one or both the copies of this gene will cause thalassemia. This will cause what? Thalassemia. Fine, right? Cystic fibrosis. Okay, here 65 roses. Like that you can remember. Mostly students will be remembering like that. So, inherited by what? Autosomal recessive manner only it is inherited. This is because of the gene which is affected on the chromosome 7. This is a genetic disorder which has direct effect on lungs, where the complete fluid is packed in the form of cysts in the lungs, and this affects breathing and leads to suffocation for the patient. So, here you see cystic fibrosis is a genetic disorder, this affects mostly lungs, but also this has its effect on pancreas, liver, kidneys and intestine. Unlike film, the survival probability is 0.1%. Hardly people will survive who are having cystic fibrosis. It is very difficult because it has its effect on every part of the body. Okay, primarily lungs will be affected. So, here you see long term issues include difficulty in breathing, coughing up mucus as a result of frequent lung infections. The mucus traps germs and this leads to infections. It can also cause severe lung damage like cysts. So, what are cysts? Fluid filled sacs and fibrous, these are scar tissues. Fine, right? This is about cystic fibrosis. So, what are the 4 types of disease that we have exercised? Autosomal recessive disorders. First is sickle cell anemia, thalassemia, phenylketonuria, cystic fibrosis, right? Now, here chromosomal disorders. Look at it carefully. Okay, I will anyhow send you the PDF later. Chromosomal disorders, as I told you, either it can be change in the chromosome structure or chromosome number. If it is on the chromosome number, that is addressed as aneuploidy, which causes non-disjunction of pairs of homologous chromosomes. What happens is, for example, if this is the transmission of the chromosome, non-disjunction of the chromosomal part and transmitting only half of the part like this. This is non-disjunction of the chromosomes. Fine, right? As likewise, it can be trisomy, means along with a diploid set, additional chromosome also you are getting. This is trisomy condition. Monosomy means only half of the chromosome is getting into the cell, that is monosomy. Trisomy is caused by addition of one extra chromosome. Monosomy, deletion of one chromosome. Nullisomy, both the chromosomes will be deleted. And polyploid condition we have learnt in cell cycle only, where polyploid condition is resulted because of sequential karyokinesis, which is not followed by cytokinesis. Karyokinesis continuously, this is not followed by cytokinesis. that results in what condition? Polyploid condition. Chromosomal disorders. We have 3 disorders, very important: Down syndrome, Klinefelter syndrome and Turner syndrome. Down syndrome is because of extra copy of chromosome number 21. This is discovered by John Langdon Down in the year 1866, where individuals with Down syndrome will bear 47 chromosomes, 45 plus XY in males, 45 plus XX in females. One extra set of autosome will be there. This is the karyotype. Okay. Can you see extra X chromosome on 20? So, this is the karyotype of Down syndrome. Look at the symptoms, very important. Quite a times the questions are put on the symptoms of Down syndrome. If you see, most children will show facial features, like they will have downward slanting eyelids, round flat face, tongue protruding outwards all the time. They will have mental retardation, poor skeletal development, smaller skull, palate is arched like this all the time, palate is arched, and flat hands with many creases on the hand. So, these are the symptoms of what? Down syndrome. Next, if you see with a Klinefelter syndrome. Klinefelter syndrome is observed in males. This is because of inheritance of extra X chromosome. Okay. Here can you see in the allosome XXY. This extra X chromosome will add few more feminine features to them, to their stature. So, if you see the extra X chromosome, what is it giving? What is the symptoms of Klinefelter syndrome? Remember all the time, Klinefelter syndrome observed in males, not in females. Okay. So, here development of breast or gynecomastia is seen in males. These individuals are tall, thin, they have poorly developed secondary sexual qualities, because masculine features are dominated by XX. Fine, right? Next, spermatogenesis is absent. Such individuals are sterile. They are usually having normal intelligence to normal intelligence, but the other features like secondary sexual characters will be affected. Few of the feminine physical features are present in this people who are having Klinefelter syndrome. Next is Turner syndrome. Turner syndrome is observed in females. Fine. This is absence of one X chromosome. They will have 45 plus X chromosome, that is it. It is a genetic condition in which female is partially or completely missing on the X chromosome. They lack the X chromosome in females. This makes the karyotype condition as 44 plus XO. Fine, right? XX chromosome. Now, what are the symptoms of Turner syndrome? Fine, right? Here females with Turner syndrome, of course, they are sterile, their ovaries are rudimentary, they lack secondary sexual characters, no secondary sexual characters, they have short stature, webbing of neck and low positional hairline. These are the features of what? Turner syndrome. Clear right now? Next, pedigree analysis. Before looking into the pedigree analysis, you should actually know what is pedigree analysis. Okay. Looking into the complete family chart and their pattern of inheritance, we can identify their disease and pattern of inheritance of the disease. So, making them completely visible in a single chart is known as what? Pedigree analysis. Analysis of trait in several generations of a family is called as pedigree analysis. You have to know the symbols and shapes clearly that we are using in a pedigree analysis. A symbol square box is what? For male, female, sex which is unspecified. This is for carrier female, unaffected males or individuals, these are affected individuals. Normal mating, fine, right? This is parents normal but affected individual. If they are dead, this is the thing. Mating with relatives, this is consanguineous mating, one time asked PYQ. Next, parents and children's below, unaffected. Fine. So, this chart I have already dealt. Autosomal recessive, tell me what are the diseases? Phenylketonuria, cell anemia, thalassemia, cystic fibrosis. Here X-linked recessive, hemophilia. Good. How can you identify autosomal dominant and autosomal recessive? First you should know the combination of the alleles. Autosomal dominant is either this or this. Fine, right? So here if you see heterozygous individuals, fine, right? Whenever autosomal dominant allele is there or autosomal dominant disease is there, how you can identify this on the pedigree chart? First thing, there will not be any skip of generation. In every generation you can see the disease. Second thing, affected parents can have unaffected offsprings. F1 generation, the affected parents can have unaffected offsprings. Okay, and affected individuals are always, at least they will have one affected parent. Here you see affected individuals, at least one affected parent. Affected individuals with at least one affected parents. Fine, right? These are the key features where 80% you can confirm that is autosomal dominant. After confirming, you have to apply the alleles and see. Then it goes correct. Fine. You got the features like how you can identify autosomal dominant. Next. Autosomal recessive. Skip of generation will be there. Autosomal recessive. Here, unaffected parents can have affected children. Unaffected parents can have affected children. There will be skip of generation. Fine, right? So, here you can see in this though there is no skip of generation, but usually in most of the things skip of generation will be there, and unaffected parents can have affected offsprings. Because for example, if this is the genotype of the parents, what are the possible combinations of the alleles? Fine, right? They are having affected offspring, no? Likewise. Next. X-linked dominant, whether it is dominant or recessive, allosomal, autosomal, there will not be any skip of generation. Okay, there will not be any skip of generation. And here all the daughters of affected father will be affected, because affected father will have what? For example, this. Then daughters having this will be affected, no? Let us suppose I am telling. So, heterozygous condition also will give the effect of the disease. So, all the affected fathers will have affected daughters. Okay, and there is no skip of generation. Disease will never transform from father to son, because X chromosome of father is affected. From father the gene will not go to son, right? Y chromosome is inherited likewise. Fine, right? X-linked recessive, of course skip of generation will be there. Carrier female will show the expression of the disease on the males. Carrier females will give the disease to sons. Okay. So, here it will show criss-cross pattern of inheritance. So, what are the features, ma? First is skip of generation will be seen. Carrier females carry the disease to sons. Okay. Third thing, criss-cross pattern of inheritance. For example, grandmother, from grandmother the disease is travelled to son to granddaughter. From grandfather the disease is carried through female through to grandson. If the disease is travelled through male, that is addressed as diandric. This is addressed as diagenic. Okay, likewise. So, it is showing what? Criss-cross pattern of inheritance. Clear till here? Any doubt anywhere? Understood now how to identify pedigree? Now, we will move on to PYQs. Look into that, answer. Hmm? Look at it properly and answer. Carrier female, if you can see, you can go for X-linked recessive, because in X-linked recessive only you can see carrier female. What is that asked in the question? With the help of given pedigree, find out probability of the birth of a child having no disease and being a carrier. In which generation? F3 generation. In F3 generation, you have to identify having no disease but a carrier. Okay. Let us suppose this is what? Cross between carrier female, affected male. Do and tell me. What will be the probability? One-fourth of total offsprings. One-fourth of total offsprings will be affected. Will be carriers, sorry. One-fourth of total offsprings will be carriers. How many will be affected? Half. How many will be unaffected? One-fourth. One-fourth carriers, one-fourth unaffected, half affected. Look into the combinations, do the cross and you will get to know that. Simple it is. Next question, simple. Good. Shakil Khan, very good ma, continue. Hmm? 9:3:3:1, good. This question, polygenic inheritance. What is the pattern of polygenic inheritance? Non-Mendelian inheritance pattern, because that is an exception for Mendelian law of inheritance, right? Which is option 4. Good. Next question. Sickle cell anemia is autosomal recessive. He is saying both male and female are heterozygous. Let us suppose this is the condition. What is the percentage of progeny deceased? Both heterozygous means only one will be of this type. No? The probability of effect is 25%. Next question, good. Simple. Next. Don't get excited, questions will not be that simple. Identify the wrong statement with reference to gene I that controls ABO blood grouping. Yeah, wrong statement. No. How come IA and IB will produce same sugar, man? This is wrong. Next, select the correct match. Very simple. Odd man out. This one means, tell me what is the right answer. Thalassemia, recessive disorder. Hemophilia, X-linked recessive. Phenylketonuria, autosomal recessive disorder. Very good. Next. Experimental verification was done by Thomas Hunt Morgan, right? Look at this question. As pairs he is mentioning, as pairs which were similar except in one character, for contra it is 14. Good. Simple, the one which we have dealt. If a colorblind female marries a man whose mother is colorblind means he is affected, right? And colorblind female. What are the chances of her progeny being colorblind? Do and tell. If a colorblind female marries a man whose mother is colorblind, what are the chances of her progeny having colorblindness? Good. Yeah, it is 100%. Very good. Option C. Next. Always in the gene mapping you have to go from maximum to minimum. Maximum gene recombination frequency means maximum distance. Maximum distance is in between what? A and D. So A and D are at the extremes. With what? 29% of distance or recombination frequency between them. After which C and D. I can plot C here, which is 24%. After which A and B. I can plot B over here, which is 20%. Followed by BC. B and C how much ma? 15%. Followed by B and D 9%, A and B 5%. So it will be A, C, B, D. It will be easy. You have to go from maximum gene distance to minimum gene distance. You can plot it easily. Okay. So it is A, C, B, D. This one. Which of the following occurs due to the presence of autosome linked dominant trait? Autosome linked dominant trait. Finally, try out this. Good. XO, grasshopper. Last question, Abdul. Statement 1 is true. 2. Both are correct. Statement 1 and statement 2. Good. Got some confidence doing PYQs? Exercise it, revise it multiple times, practice as many objective as you can. Okay, then you will be fine tuned with this chapter. In our next class, we will be starting with mol bio. Okay, yeah, that's all beta.